Welcome. Thank you.
All right. So this is a work in progress. I'm not finished. And so I don't know the entire how to do a thesis project yet, but I know the beginning, which is probably what is going to be most helpful for you guys. Let's see. So getting started, I think the best thing to do is to just read a lot about the project without really having any ideas in mind, like letting the ideas come to you, figuring out what has already been done, and like not trying to do anything, just letting your mind be open and reading all these things. And a great place to start with that, if you have access, is Google Scholar, because it only shows you things that are publications or books. So it can filter out a lot of the silly things that you can get on just regular Google. So that's really helpful. And I'm going to show you some useful tools that Scholar has. For example, if I were to go here and look up overwintering honeybees, like I did 100 times, if you want to cite something, you can click on these two little quotation marks, and it comes up with all these different types of citations. And I like to do APA, but you can choose whatever. And if you just click on it, you can copy and paste it and bring it over to your working document. So after after you've done a lot of reading, and while you're doing a lot of reading, I like to make an annotated bibliography. I think they're really useful for taking notes while you're reading, and for later reference when you're thinking about your project, writing up your results, coming up with questions. And while you're reading in your annotated bibliography, a good thing to do is to summarize the paper, what they did, what they found, the things that were actually their findings and not like what they were referencing. So you also want to like look into who the authors were, what their credentials were. And if you have any questions about the papers, you can email those authors, ask for questions, other information, or just to talk to them, because they probably would love to have a captive audience. And it can be really good for networking and things like that. So I'm going to share an example of my annotated bibliography that I made for this project. And it's, I think, like 10 pages long. I just put the reference for the paper and then a little blurb about it, so that later on I can go back and reference it, find where things were, where papers were, so that I can cite them later when I'm writing the paper up. And it's really great to use like the Command F tool. So if I want to know which papers we're talking about juvenile hormone, I can do that and go through. And it's a really great resource, so you can see what's been done. And you'll find when you're reading these papers that sometimes you'll have some questions about them that you go down another path to read more papers to figure out so you have some question in a paper and you go read another paper, and then you read another paper because you have a question from that paper, and then eventually the ends sort of get tied up, so you're not getting any new papers anymore. And that is when you know you're done reading papers, when you're not getting any new information. So yeah, I had to read probably 50 to 100 papers before I got to that point, when the ends start getting tied up. So that's when you know you're done. And you're probably not really going to be done, but that's when you can sort of slow down. And so while you're reading these papers, you should ask questions, synthesize all of the information that you know, and figure out how it all connects. Look for holes in the literature that we haven't answered yet, and talk to other people once you figure this stuff out. So this is an example of what I made when I had all this information that I didn't know how to put together. And you can see that it's really pretty complicated. And I not really expecting to try to explain it to you guys, but I think it's interesting to see how all these different things connect and how they came from all these different papers. So briefly, overwintering honeybees live for a lot longer than summer honeybees, and we don't really know exactly what causes them to enter this different state. We do know that they are influenced by the environment to some degree and by hive demographics to some degree. And we also know that winter honeybees they resemble nurse bees in certain ways. And so it's kind of like they're in this fixed state of eternal youth. So when I'm asking questions about this problem, the overarching question is what factors contribute to the transition to overwintering? Because we don't quite know exactly what it is. But then to get more specific, to come up with a question that you can create a testable hypothesis for, I asked the question like, how does a particular pheromone, ethyl oleate, affect the physiological markers of overwintering? And so a little bit of more background about them is, like I said, they live long-lived. They live about 4 months, whereas up to 4 months or somewhere around there on average. And summer bees only live about 6 weeks, the workers. And we don't know what triggers it, but some of the environmental factors could include cold or temperature, day length, pollen dearth, rain, those types of things. Sociodemographic cues could be pheromones, caste division, the number of bees in each caste, things like that. And we don't know how they all interact, probably a lot of different interactions coming together. But I'm interested in looking at how you could maybe use the effects of overwintering to your advantage, how to prepare for overwintering, things like that. So the way that winter bees resemble nurse bees is that they have increased longevity, obviously, but they also have increased hypopharyngeal gland size, which are the glands in the heads of the bees that are used to create brood food. They have low juvenile hormone, which is a hormone that influences flight. So older bees have low juvenile, or they have high juvenile hormone, whereas young bees have low juvenile hormone, which is a little bit counterintuitive because it's called juvenile. They have high vitellogenin, which is the hormone associated with yolk development and fecundity. And they have larger fat bodies, which is something that's going to help them live through the winter on reduced stores. And as many of you probably know, in the winter, they usually cluster and they don't lay eggs. And the queen is still present, but she doesn't lay eggs during that time. And they have a decreased metabolism because metabolism, if you're going to be metabolizing your food stores really quickly, you're going to not have much to last you through the winter months. And metabolism is associated with flight, so they're not flying either. And the thing that distinguishes winter bees from regular nurse bees physiologically is that the hypopharyngeal glands are not actually active in producing brood for the colony because there is no brood. Brood pheromone is required for the hypopharyngeal glands to activate. And so they're really enlarged, but they're empty. And so that's one physiological marker that we'd be interested in looking at with fat bodies. It's something that sets winter bees apart from any other bee, is that hypopharyngeal gland state. And there is some evidence that young foragers can revert to a nurse state when necessary, depending on the sociodemographic conditions. And they have an easier time reverting than older foragers. One of the experiments that was done about that was they took all the nurse bees out of the colony and left the brood, and the bees that had already been foraging started foraging again, and they developed hypopharyngeal glands and things. So they were able to revert if they were, if they had to. And so the question kind of for me is, can older bees revert to a nurse-like state, or is it only nurse bees that become winter bees? And finally, or this might not be finally, ethyl oleate causes nurse bees to stay, this is a pheromone that they produce. It causes nurse bees to stay nurse bees longer, and it indicates that enough foragers are present. So if you have a bunch of bees returning to the colony with honey, it tells them we have enough bees coming in that are foraging. We need more bees to stay as nurse bees. And the way that they do that is with this pheromone called ethyl oleate. And ethyl oleate is produced when foragers consume fermented nectar and transfer it to nurses. And the question that I have about ethyl oleate specifically is if the bees are staying in their colony longer because of poor weather conditions and things like that, if the ethyl oleate concentrations may be higher, causing them to sort of revert to a nurse-like state. And so I'm wondering if ethyl oleate could influence a transition from a summer bee to a winter bee. And so that's like the basis of my project. So when you're designing an experiment, it's important to be realistic and to not overextend yourself. And you can always change it as you go if you realize that that was kind of crazy. So first you want to start with an annotated bibliography and your introduction. So what is the background information? What is already present in the literature? What do we need? And you want a hypothesis. So for my experiment, the hypothesis is if ethyl oleate contributes to late summer, early fall transition to an overwintering state in honeybees, then exposure to ethyl oleate will cause young foragers to change their physiology to resemble winter bees. And that's just your hypothesis. To go further on that, you want to have your predictions, which is is a little bit more specific. What do you actually expect your results will be? And it's fine if they get proven wrong. It's going to be interesting either way. So hypopharyngeal glands, in my case, the predictions are that hypopharyngeal glands will enlarge, but they'll remain low in protein when they're exposed to ethyl oleate compared to when not exposed to ethyl oleate. The fat bodies will get larger, and these effects will be more prominent in young foragers than in old foragers, and possibly non-existent in nurse bees. Or it'll have, instead of a drop, it'll just remain low or high, depending on which effect we're looking at. I had two methods of administering the ethyl oleate in my experiment because this kind of experiment hasn't been done that often, and I wasn't sure what would be the most efficient way and what way ethyl oleate would be perceived by the bees. It's supposed to be a volatile chemical, but we weren't sure that it would be attractive to the bees, so we also wanted to feed it to them in case they weren't even going near it. So I had a glass plate and a sugar mixed in with sugar. So there's one where I just put ethyl oleate on the glass plate and put it in a cage, or there's one where I mix it in with their sugar candy. So I had two different treatment groups there and two different control groups there. So one of my predictions would be that the glass plate and the sugar plate wouldn't really be any different, but we needed to cover our bases. And we also found from the literature that we wouldn't expect much of a result until after 3 days of exposure to ethyl oleate. The variables that I'm looking at in this experiment are hypopharyngeal gland size, longevity, gene expression of particular genes, and fat body content. And I would encourage you, as you're working on these types of projects for yourself, to write as you go so you don't forget what you've done and you don't get confused with the literature. And like I said, don't be afraid to exchange your experiment as you gain new information. There were a few other variables that I was interested in looking at that I wasn't actually able to do ultimately because it became too big for my project. And it's really great to kind of divide up the work before you even get started by thinking about the sections that you're going to need to cover, and like dividing it into your hypothesis, your predictions. It kind of organizes your thoughts and keeps you going in the right direction. So parts of a paper that you need to think of if you're going to be writing a paper is your abstract, which is your summary of the whole paper, which you should probably write at the end since you need to know your conclusions and things. Your introduction, which you can write really early because it doesn't matter what your results are. Your methods and materials, which should be as detailed as possible because they need to be repeatable. Your results, the discussion, how you synthesize your results, how you interpret them, their greater importance to the field, etc. And your final conclusions. And then your references, which you could pretty much just harvest from your annotated bibliography if you do the work early then. So that's really great. And then, the scientific method is, most people probably are familiar with it, but you ask a question, you do your background research, you construct your hypothesis, then you come up with an experiment, you draw conclusions, and then you determine if your hypothesis was true or not, and then you report. And then you do it again if you have more questions or something. And usually the more research you do, the more questions you have and the fewer answers you actually have. So that's fun. So my experimental design was I painted emerging day-old bees from a single colony so that they would all be related. And they emerged in waves. So I ended up having to assign a different color to each day. Ideally, I would have had all 6,000 bees or something emerge from one colony and paint them all in one day, but that was just logistically impossible. So I painted them a different color for each subsequent day. And then I inserted those painted bees into a colony in a tent. The purpose of that was to reduce drift and loss of bees. I didn't want all my hard work painting bees to just be lost if all of these drifted to other colonies in the yard. So I put a big tent around them so that they couldn't leave. And I don't know if that had an effect or not, but hopefully not. And then I removed a subset of those bees from the hive on the 7th 21st, and 28th day and put them into cages separated by treatment. And the purpose of those days was I figured that on the 7th day, they would still be nurses. On the 21st day, they would still be relatively young foragers. And on the 28th day, they would be considered old foragers. Unfortunately, when I went to collect young foragers and old foragers outside of the hive, they weren't really foraging, maybe because of the tent, which resulted in me having to pluck them directly from the hive. So I don't know whether that would have an effect, because more than age, task actually influences your like behavior and physiological effects, especially in relation to the parameters that I'm interested in studying. So here's a couple of pictures to sort of illustrate what I did. I painted the bees, I painted the bees in the bucket. And so on the first day they were green. So you see here, they're all green. And then I inserted them into this tent, which you can see on the right. And there was a colony in there, you can't see it here. And on the upper picture, you can see that there's all my different colors of bees together on one frame. So I take the frame out and I'd pick only the blue ones on this specific day if I needed it. And there's the colony inside of the tent. There's a little chicken sugar feeder. So you can see that's what I wanted them to feed off of. They actually weren't that interested, but I wanted to encourage them to forage, but it didn't work. So that's why I say that you should just roll with the punches if you need to. So to illustrate what I did here, on the first day, I got around 1,000 bees and I painted all of them green. And there was like a donor colony and the colony in the tent. So the first day there was 1,000 bees, they were green. The second day there was 1,000 pink bees. On the third day there were 1,000 yellow bees. And on the fourth day there were 1,000 blue bees. So the only difference between the color groups is that they emerged a day later, and it shouldn't make a difference, but I needed to keep all of these parameters separate until the very end of my experiment so that I know that the day that they emerged isn't a confounding factor. And they were all moving from one colony to another colony. So there was, 4 different colors. There's actually 2 other color groups, but I'm not going to include those in this because it gets confusing. So then on the 7th day, after 7 days of being in the colony, they're 7 days old, there should be nurses. I took out 200 bees, 200 of the 1,000 bees, and I divided them into 4 different cages. And there were 50 bees of each painted color group in the 4 cages. And they were divided into the ethyl oleate administered on candy, ethyl oleate administered in paper, control candy, and control paper. So that's just blank. They're just a piece of paper in there with nothing on it. And I actually kept the ethyl oleate groups separate in a separate incubator because I wasn't sure if it was volatile, which it's not supposed to be, it could kind of permeate the entire incubator. And so control groups would be getting treatment when we don't want them to be. And in addition to the 50 bees, I also added 2 tablespoons of day-old emerging attendant bees because 50 bees is not enough for the bees to feel normal and cozy. So that was what I did on the 7th day that the green bees were in. On the 7th day that the pink bees were in, I did it again. On the 7th day that the yellow bees were in, I did it again. And then on the 7th day the blue bees were in, I did it again. So you can see that ends up being a lot of cages. That was 20 cages for that group. But then on the 21st day, I did it again. They should be young foragers. They're in quotation marks here because they're not actually foragers, but that's what I called them throughout the experiment because that was my initial intention. They're really just 21-day-old bees. And I did it again. So I did it with 4 different colors on 3 different treatment groups and 3 different age groups and 4 different treatment groups. So it ended up being something like 60 cages. So you get the idea there. And I did it on the 28th day as well with the old foragers. So I ended up with 60 cages, which was quite a lot in the pink. So what I did once they were in those cages is every day I would go in and take out the food that they'd been given the day before and weigh it to see how much they'd eaten since the day before, both pollen and sugar candy. I fed them new candy, give them new treatment, and I counted the number of painted dead bees that were in the cages. And so since everything happened like a day apart, everything ended up being staggered. So it ended up being quite a lot of work. per day, even though when I designed the experiment, it didn't seem like it was going to be that much work because I thought, I'll only come on the 7th and 10th and 14th day, but it ended up being a lot more than that. You also don't realize how much work feeding 60 cages is going to be. It seems like, oh, I'll just go in and feed them, but then you end up being there all day just taking out because the bees would try to escape and it was just a mess. But then, so once they'd been in the cage for 7, 10, 14, or 18 days, I would, so on the 7th day, I came in and I sampled 10 of the colored painted bees and put them on dry ice for later analysis. So I did that on the 7th day for each cage, the 10th day, the 14th day, and after 18 days, I had run out of bees. And I also actually had another group of painted bees that I didn't do anything with so that I could count how long they had lived for without sampling any of them to get survival data and determine how long the different bees lived for. And I was originally going to collect hemolymph from each of these bees and analyze juvenile hormone, but it took too long and I didn't expect that it would yield very many valuable results. So I decided that I would keep that for maybe another project because it just ended up being too much work. Just like not even enough time in the day. So far for the analysis, I've dissected the hypopharyngeal glands out. I sent them to the proteomics center to see if they have that brood food in them. Like I said, if they're winter bees, I would expect them to be big and that they would be empty. So I need to determine if they're empty or not, which is what I'm waiting on. I'm also going to be looking at the gene expression for one gene that makes alpha beta hydrolase enzymes, which are what metabolize ethyl oleate. So I want to see if exposure to ethyl oleate allows you to metabolize it better, it makes you more sensitive to it or not. And then I'm also going to be looking at fat body weight and size to see if the bees that are exposed maybe have greater fat reserves, more ability to overwinter. So here's just a couple of pictures. These are the cages that I was working with. As you can see, I had to open this little sliding door at the front, and the bees would just all try to crawl out, and I needed to prevent that from happening as much as possible. And they had these little caps filled with pollen and sugar, which I would mix ethyl oleate into, which is just, I think it's an oil base, so it's like a clear liquid. And then they also got water in these little Eppendorf tubes. So yeah, there were 60 of these cages. Now, the hypopharyngeal glands are these glands in the head of the bee. They look like a nerd's rope, and they produce the brood food for bees, for the babies. So basically royal jelly is what they produce. And this is a picture from under the microscope on the left is the face of the bee has been removed, and you can see that the hypopharyngeal glands just sit right on top. And these ones actually have these little black specks on them, which I think might be the result of consuming ethyl oleate, which they're not really supposed to do. I think it may have caused them to sort of atrophy or hemorrhage or something. So every now and then I would get these bees with these weird little black dots, and I don't know what that is. And so the way that we analyze the size of the hypopharyngeal glands is I took these pictures all at the same magnitude, so you can see really here the stark contrast between the sizes that you can get between different bees. The one on the right they're pretty small, they're kind of shriveled up, whereas on the left they're really big and full. And we used an image software called ImageJ, and I had an undergrad do all of these. We have over 800 pictures of hypopharyngeal glands. And she went through and the protocol here is to circle the individual acini, these little bubbles of the hypopharyngeal gland, and you're supposed to do 10 of those per photo, and you get sort of an average size, and then you use the software to tell you what the area of that circle is. And so we're going to be comparing between treatments, the overall size, and then we'll be looking at the protein content as well. So some of the results that I acquired, I wanted to see if I needed to prove that they were maybe consuming the ethyl oleate in the candy or whether it was attractive to them. And it looks like when they were fed candy, if you compare like the blue bars to the yellow bars, there doesn't seem to be a huge difference across the treatments between consumption of candy. So they ate it whether or not it had ethyl oleate in it. But there seems to be a bigger variance in how much pollen they consumed. So if you look at the nurse bees, there's a pretty big difference between the EO paper and the control paper. So it seems like if you have exposure to ethyl oleic, you want to eat less pollen. And so it would be interesting if, even if they eat less pollen, maybe their fat bodies will be bigger. So we'll have to wait on those results. And there does seem to be a pretty big difference in the difference depending on the age group that they're in. So the difference seems to be a little bit more severe, maybe in the nurses or in the old foragers, but they also just consumed overall more pollen when they were older, which is interesting because nurse bees generally need to be feeding the brood, and so they need to eat more pollen. So that's interesting. So with those cages that I said didn't have any bees sampled from them, I was able to just get straight survivalship data. And the difference between the treatment groups in the nurses is not that big, but as you get into the young foragers and the old foragers, that difference widens. And so if you see at the old foragers, which is where the biggest difference is, the blue, which is the EO paper, and the green, the control paper, there's a pretty big gap there. And it looks like the EO paper, they survive longer when they're older, which is contrary to my hypothesis, but still quite interesting. I expected the young foragers to have the greater effect from the treatment, but the old foragers did. And that might have to do with the fact that they weren't actually foragers. They were just older. And so these are the results from the hypopharyngeal gland size, the acini size. The first dot on the graph at the 0 mark on the x-axis shows the value for the hypopharyngeal glands if they were bees. When I was sampling the bees out of the original colony, I also took a subset of those bees and didn't do anything with them and killed them immediately. So that's a value that shows the before. So with the growth, it's the before minus the before, so it's 0. And you can see how much the hypopharyngeal glands have grown at each sampling date. And it's sort of confusing because they all shrank. So it's basically like which one shrank the least. So if you look at day 7, let's see if I can get my little pointer here. I wasn't able to get that a minute ago. I can try to point on it. Can I do that? Oh, yeah. All right. Well, I drew a little line. Here we go. Undo. All right. Can you see my pointer, Wendy?
Yes, I can.
Okay, so on day 7, we have the EO candy, which is at the bottom. That one is the one that grew the least, so it shrank the most compared to the original measurement. So we want to look at, for example, the control candy. It actually stayed the highest for the longest, but then at the end it kind of switched and the blue got better. So it is interesting at the point when they're all still decreasing, but maybe they decrease a little bit less. Or in this one actually like kind of recovered, which is cool. And so like the difference between those is, I guess, greatest in the young foragers. I haven't really analyzed this data yet, so forgive me, but you can see how there is a difference between the treatment groups, and the difference depends on the age group. And so those are pretty promising results, although I haven't fully interpreted them yet. What's left for me in my project is the protein analysis, determining if the size and the activity of the hypopharyngeal glands are correlated. And that'll be a really big diagnostic for whether ethyl oleate creates winter-like bees. The gene expression for the alpha beta hydrolases, which is quite a long process. I have to first extract RNA, which is extremely sensitive, then remove the DNA so that nothing downstream is acting on DNA, do reverse transcription, which is to create complementary DNA, and then doing qPCR and possibly RNA sequencing, and then doing the fat body content, which will hopefully be much simpler than the gene expression. So getting back to how to create your own experiment and some tips that I have for doing your write-up. Write your methods as you go so you don't forget what you're doing. They can be really detailed, and explain your steps. So when I said that I was putting my bees in a tent, I want to also say that the reason behind that is because I wanted to reduce the loss of painted bees and drift to other colonies. If I just said that I put it in a tent, people coming along in 10 years and reading my paper would be quite confused as to why I did that. Be as detailed and clear as possible while still being concise. You don't want it to be really, really long and rambling. Write your results as you acquire them, because if you're trying to remember what you did and figuring out what your results mean and creating your results and everything, it'll be a lot of work if you leave it all for the end. So it's kind of like when you're cooking, you need to clean as you go. And also you can write your introduction before you even begin your experiment. And that's probably even better because it would be less biased and you'd have all of your literature kind of simulated together. And so you can do that before you even get your results, and they should be independent. And I'll show you briefly of what my results look like. So here I have my methods, and it's really pretty detailed. I talk about, like 70% powdered sugar to 30% corn syrup to create my bee sugar. And it goes into quite a lot of detail, but it also is not that long. And then the treatment is here. These are all the different things. So I've written them as I'm going, and that's as far as I've gotten. So I've pretty much just written my methods, but I also have some introduction in other places, like my thesis here. I have this long introduction that references all my references, which is important, and it's easy because it's already all organized by who said what. So that's why I really like having this annotated bibliography here. So yeah, that's pretty much it. If anyone has any questions, please feel free to ask.
Thank you, Nissa. That was great. And I love your needlepoint at the end. Think it over, think it under. Yeah. Did you create that?
I did. And the quote is actually A.A. Milne, the author of Winnie the Pooh.
Yes. Yeah. Yeah. That's wonderful. We do have a couple of questions. And I invite Holly, would you unmute yourself? And I invite you to ask Nissa your question.
Oh yeah, sure. Let me turn on. Okay, I wanted to know, because I'm in San Diego and we just had a storm this weekend and I knew it was coming on Wednesday. So I did, fill up their Vivaldi boxes with some sugar, and I had hoped I wouldn't have to go in again for a while. The temperatures dropped like 20 degrees, but still there were a lot of brood in all of I've got 4, and I anticipate that when it warms up in another week, they'll be flying again. So I'm just wondering if there's a continuum between those differences when you're talking about the winter bees in the north versus in the south, because in the south, they do forage almost year round here, and it certainly can do cleansing flights and all that. So I just wonder if you are focusing on true winter bees.
Well, so actually when I started here, Dr. Nino gave me a few options of like ideas she had of what things we should do. And one of the questions that she was really interested in was, do we have overwintering bees in Davis or in San Diego? And I started doing some research to try to figure out how I would test that question. And there was so little known about how wintering even works in honeybees at all, even in the north, that it was like, how was I going to do a project on whether we have winter bees in San Diego if I don't even really know what a winter bee is? And so it kind of led me down this rabbit hole of, well, how do we tell what a winter bee is? And so or like what causes winter bees? Like if it isn't temperature and day length, then maybe you could have overwintering bees in San Diego, depending on, dearth, for example, is something that I've thought about, because there seems to be like similarities to winter bees in tropical regions, places like San Diego, places like Brazil, or like during times when there's no pollen or when it's raining a lot. So I don't have an answer for you other than that I went down this rabbit hole, and the question really ended up being, what even is a winter bee? And that's awesome, like really a great example of how research works. I started out with that question that you just asked me, and I ended up here. So I think there's a lot that still needs to be done to be able to have like, big field, like application level experiments about how do we really manipulate overwintering as beekeepers, because we still just don't understand it very well at all.
I think that's great. And I just wanted to add that when you were going through the presentation of all the steps and all the things you were, the different days where you were pulling bees out and then all the days you were feeding them, and you were like, it was actually a little bit, quite a lot of work. I was like, I didn't want to interrupt, but I wanted to say, it seems amazing and incredibly overwhelming, the amount of work you must have done. So I just appreciate it and I'm very impressed.
Thank you. Any more questions?
Thanks, Holly. John, would you mind unmuting yourself and asking your question? John Winsler has a question regarding, he said, could you tell us again how you determined the overall size of the hypopharyngeal gland?
Okay, so
Yeah.
first I dissected the hypopharyngeal glands out under the microscope by taking off the faceplate and then removing them. I dissect in what's called bee ringer solution. It's the solution that's isotonic to honeybee hemolymph. So it's the same basically composition, salt levels, so the glands don't like dry up or explode. And so when I take them out, it comes out pretty easily. You just sort of pull it and the whole little strand comes out. And so when I do that, I take a photograph. We have a microscope with a camera attached to it. I take a photograph and I like calibrate it by taking a photo of a small, of a very small ruler, and then I can calibrate the data there. And the image the software that I use is called Image J, and I can put that in the chat if you're interested in doing something like this. And it's free and you can just download it. And when you go in, you use a little ruler in it, you measure your ruler, and then you tell it like this many pixels equals 1 millimeter or something. And then from there, it can remember that and do all the calculations for you. You basically just draw a little circle, you trace the size of the hypopharyngeal gland, and then you hit measure and it tells you the area. And the way that you do that is, or the overall protocol is, you circle 10 different acini, these little bubbles. And you're supposed to try to get them sort of evenly dispersed across like a variety of different areas of the hypopharyngeal gland, so that you're not getting any regions that are particularly decayed or something. So they're kind of scattered, as you can see. And then we take an average of the area for each of these hypopharyngeal glands. And sometimes it was pretty difficult because, as you can see, like areas of the picture on the right, some of it is kind of blurry and you can't always see exactly what you're doing. Some researchers, when they don't have to do protein analysis at the end, will stain the hypopharyngeal glands using like a blue dye that makes it more visible. But I couldn't use that because I needed to keep them like clean for downstream analysis.
You're doing the same thing for fat bodies?
Fat bodies is actually a different process, and I haven't done it before. It involves like using, and I haven't really researched it yet. Eventually I'll figure that out, but it involves using a freeze dryer. I think you have to freeze dry them, and then you take the difference between the weight that you had before you did it and the weight you had after, and then you can extrapolate that was the size of the fat body or something like that. I'm not entirely sure, but a lot of these questions, if you do your research, they'll probably will have like protocol papers, like full papers that just tell you the steps to do to take it. In fact, there's a pretty simple little paper by, I think, Vanessa Corby Harris that explains how to do these hypopharyngeal gland measurements really simply.
Could you theoretically do the hypopharyngeal glands with some sort of radioisotope and then measure it after the fact and compare that? Or, I mean, it's out left field.
Probably. I don't know how to do that. I'm sure that it's been done.
All right. Thank you.
Mm-hmm. Any other questions?
Yeah, thank you, John. Good question. I have a question. You've got a pretty extensive annotated bibliography. Like you said, you read between 50 and 100 papers, right? I'm curious if the thought of cold storage and how that has impacted this segment of your research anyways. And do you have, do you feel that that's something that you perhaps might want to pursue and research a little more on? And would you explain cold storage for most of the folks on the call as well, please?
Yeah, so it seems like in place, I think it's mostly Washington State that's been doing a lot of the research on cold storage. They basically just put their beehives into a cold storage facility, which is essentially a giant warehouse freezer or even just a freezer. And they have found pretty good success with the like the survivorship, and I think mite levels even drop a little bit. But I think a big part of this is that's really only utilizing one factor of overwintering. It's just they're automatically just getting thrown into these cold chambers. And I think that there's a much longer process behind the transition. It's such a big change that thousands of bees potentially have to go through to become so physiologically and behaviorally different that just throwing them into the cold storage, they may not be ready, they're not fully, it's like a very continuous process of the seasons are changing, there's less pollen coming in, the temperature is changing, the days are getting shorter, and I think that they may not be reaping the full benefits. Perhaps it's not helping as much as we think, maybe it's hurting some. I think that determining other factors that influence overwintering behavior and benefits the bees is really important to maybe pair with cold storage. So for example, if ethyl oleate does have an effect and we figure out a way to use it, we could throw a little bit of ethyl oleate into the colonies, or determining like how far in advance to putting in cold storage do you need to prep and prime your bees to really be able to take advantage of the cold storage. If you just throw them in, they may not be ready. They may not have their fat storage. They may not, all these things. And each of these tiny little questions that I just listed, that's like a year's worth of work in terms of a project. So I don't know if I will get to it, but maybe one of you will.
Yeah, that would be pretty incredible. And that also begs a question for me, because California, with all the almonds, what is that transition like coming out of thermoregulation, coming out of cold storage, let's say, or just being loaded on a truck, say in, say, North Dakota, for example, right? And they're coming out of a potato barn and boom, all of a sudden they're in an orchard in California. It's time to wake up. I'm wondering what goes on in the hypopharyngeal glands at that point as well. What do bees have to do in order to prepare for that? How stressful is that? We know it's stressful, but I'm not sure if I've ever seen any papers talking about that either. And I was just curious if you'd come across any.
Yeah, I think that there's the whole notion of cold storage and things like that is really just now getting started on being researched. And I think they've found, some success with it. But like I said, this is pretty much untapped territory. A lot of the papers that I was reading were from like the 1980s, 1985, and even papers that were written in the like 2015 that were referencing these papers didn't really make huge leaps. And it also is it's already so specific with honeybees, but it ends up being like interdisciplinary field in itself, because you've got people studying the like field level, colony level effects of winter, and then you've got people studying like ethyl oleate in terms of how it affects sociodemographics, and you have to connect these little pieces in ways that other people wouldn't normally think to do, because the guy that's studying ethyl oleate is the guy that's he's an expert on pheromones, and he doesn't think about colony level effects of winter at all. And so then you have someone like me or you come in and try to make these connections. And so all of the information is already there. You're just kind of putting it together in a new way.
Thank you. Holly, you had a comment or a question?
Oh, yeah, just when you're talking about cold storage, are you talking about the effect of torpor?
You mean like the fact that they're like sleepy and like
Yeah.
bunched together? There are other effects I think that they found, like mite levels dropping. They have just like greater like survivorship out of cold storage in the springtime. I think that the effects would be, they're potentially going into a true overwintering state. Maybe they're not, though. I don't think people have done many studies looking at what's going on in the colonies to the bees while they're in cold storage. They're just looking at the effects after they're taken out. But yeah, like I think that overwintering itself has benefits. It kind of gets rid of all the brood, so you can't really have mites during that time. So if you have people that are bringing their colonies down south so that they don't have to overwinter, they're just working them so hard they never have a chance to like rest and cleanse themselves. It also gives them the chance of like, they have increased immunity, increased fat bodies, like lower metabolism, all these things that are beneficial. And so I don't know if the bees that are in cold storage are going to be experiencing all those benefits. You're right, they may just be tired and cold. I don't know. You may have to have them enter the true overwintering state in order to benefit from cold storage to the fullest extent.
Thank you.
Thanks, Holly. Dewey, you have a question for Nissa?
Hi, yes, real interesting study, and I thought you presented it very, very well to lead everyone through, particularly as our master beekeeper program people are thinking of their research project. I did have a kind of a specific question, and you mentioned the fact that there was a lot of research a number of years ago, and now there's some additional interest, and of course the newest interest has gone to the type of project that you're doing, looking at the primer pheromones, looking at the other gene expression and all of that was not available to the earlier researchers. On the primer pheromone, the ethyl oleate seems to be some relationship to honey that is fermenting. Is there a suggestion that perhaps in the fall of the year that is a more frequent occurrence than perhaps at other times during the year?
I don't know, actually. That's an interesting question. That would be an interesting question to look into. I know that ethyl oleate it's caused by the consumption of ethanol by the foragers, and I think there might be a slightly increased incidence of fermentation in like the flowers during the heat of the summer, but that doesn't necessarily mean that they're not fermenting it more. Maybe they're getting less ethanol, but they're able to do more with the ethanol. I don't know. Maybe that would be a good question to look into, is what are the incidences of ethyl oleate production in the hive at different times of year?
Yeah, if it's higher in the fall, that would suggest even a stronger possibility that it is in fact related to helping those newly merged bees live a longer life.
Yeah, maybe. I don't know. That would be interesting.
Yeah, thank you.
Yeah, great question. Thanks, Dewey. So many questions, right? Like you said, Nissa, you start with one thought, and the next thing you know, you have to back it up before you dive into the rabbit hole that you thought you were going to get into, because there are so many unanswered questions.
Yeah, you have to pick one. You have to pick which one you would like to do the most, which one would be the most. Like when you're designing an experiment, some people love to be working their colonies. Some people don't want to be out in their colonies too much in the summer. Like Lauren, another person in our lab, she is doing field experiments all summer. She's working the colonies. I purposefully designed my experiment to be working at the microscope during the hottest part of the summer, So like, just because a question is interesting to you, maybe you can choose the question that will suit your interests and what you actually like to do the most, your time commitment, because there's so many to choose from. You just choose whichever one you would like to do the most.
Yeah, and that's a really thoughtful approach, because you don't want it to feel like a grind. Right?
Yeah, you want to enjoy what you're doing.
Yeah, exactly. So there's so much to think about when it comes to deciding on what scientific research paper do you really want to write. So I guess picking a topic might be easy at first because it aligns with your passion, right? But then tell us a little bit about once you picked your topic. Of course, we saw, based on the slides that you shared, you have a great, more or less a mind map as to where you went, right? And then I also heard you say that you decided when and where you wanted to work, which would suit you the best. So your passion continued to be your fuel. What I also heard you say was, I didn't quite anticipate that I'd have so much work to do back to back based on, the 50 bees that were emerging each day.
Yeah.
So maybe you could give some of us future scientific paper writers some insight so that we can glean from your teachable moments that you've experienced thus far, and we can move forward and be all the wiser in how we approach our papers.
Okay. So I guess I guess my thing is I started this paper, I had 4 different, treatment groups. I had this many different bees, I had this many different cages. They were all going to emerge. When I did that assignment, when I came up with that idea, I thought, they're all going to emerge on the same day. It would take me 5 minutes to feed them, all these different things. And I didn't realize, you have to mix them all up. The bees are going to come flying out of the cages. Maybe if I'd had more time, I wasn't also working and in school and all these things, I would have designed like a mini version of the experiment with 1 cage to see like how long does it take to feed these bees, and then go back and rethink it. In terms of the mind map, I tried to, sometimes it's hard. You have an idea and then you realize it's already been done. So you just have to add that to your list of things that and that can be discouraging. But then if you end up, my idea was to basically what do we know already? I want to write a review paper. And so I tried to put everything together, be like, what is going on here? Because it would have been exciting to put the pieces together, even if I didn't create any new data, because it hasn't really been done, even within such a specific field as overwintering in honeybees in entomology. It's an interdisciplinary field, there's molecular biology, there's ecology, there's field level studies, there's things going on at the colony level, there's things going on at the individual level, there's things coming from the environment, there's things coming from the hive. And so all of this research was different coming from a totally different perspective. And so even if my project wasn't like an actual lab project where I was going out and doing these cage studies, just trying to put together this little this diagram would have been science enough, because it hasn't really been put together thoroughly. And once you try to put it together, you realize that there's stuff that doesn't fit, and that's where your project really shines through, I think.
Yeah, very, very, I appreciate how your brain sequentially works. Yeah, yeah. Dewey, do you have a comment?
Yeah, that was a great discussion of how you would narrow down to get a sizable project to use. I want to return to one other comment that you made. When you rely on only one way to try to collect the information, for example, if you're doing just a field project, or if you're just doing a lab project, if something goes wrong in the field, the cages blow over, or heaven forbid there's a fire, or in the lab the electricity goes out and you lose all your samples for whatever time you've had, you're not going to get that amount of information, that data to try to analyze. And so you made the point that you tried to keep those 2 types of interest still in the project, so that you have a greater probability that you are indeed going to have some information that you're going to be able to put down as an analysis.
Yeah, I have definitely a bunch of different components. Like I said, I was originally going to do juvenile hormone, and that just took forever. And it wasn't really too big of a deal to be like, I don't need this. I have 4 other parameters to look for. And then also in terms of separating lab work and field work, I think the intention for a project like this is doing it in a really controlled environment in the lab, and then depending on those results, designing an experiment at like a field level. For example, if I find that ethyl oleate is really effective in increasing longevity and creating these synthetic, these physiological markers as seen in winter bees, maybe I could design an experiment to see like in what dose do we administer it to colonies to get an effect, and how are those colonies impacted, or do they have greater overwinter survivalship? How long do we administer ethyl oleate for? Like how far before the first frost do we need to do it to create actual applications from the science? So there's like sort of two types of experiments, like the nitty gritty and then figuring out how to apply it at a bigger sense. So that may be my next project or something like that, or just analyzing another aspect of this project.
Excellent.
So there is a next, is there?
I think there has to be, at least for my master's, I need to have 2 projects. So I'm hoping to have it finished before the next field season so that I don't have to juggle 2 projects.
Yeah.
And then I also don't want to design another experiment until I have the results from this one, because depending on what I get, I may have different questions.
Yeah, that's wise. Well, we're just about to the top of the hour. That went by very quickly. Any final questions before we say good evening? It's 8 o'clock. I know we're all grateful sincerely for your generosity of time and insight today, and in preparing this for, well, exclusively for the camp, because this is one of our master tracks, and it's important for us folks, me especially. I've not really been down that scientific writing of a scientific paper path. So I learned a lot this evening, and as I'm sure everybody on the call did. And we're all really looking forward to hearing. We're going to have you back, because we want to know what the results were, right?
Yeah, excited to share them.
Yeah, we look forward to that. So thank you again for your time with us this evening, Nissa.
Thank you.
And thank you, everybody who has joined us here this evening for an hour. We hope you enjoyed it. And we look forward to seeing you on the next webinar. We'll be getting together again in January. Until then, be well.
Thank you. Bye.